We have recently talked about how a single-celled bacteria can do much the same things as a creature that might be made out of 3 trillion cells. And today what we're going to do is we're going to talk about how cells are able to interact and survive in their environment. And so I'd like to start off with a little bit of a scenario and kind of a brainstorm to get you thinking about this idea. So in the first setup at the top, we have a grape that was soaked in regular water for about 3 days. The second setup, we have the same similar sized grape soaked in salt water for 3 days, and we notice what happens. The regular water, the grape actually swelled and became larger. The grape that was put in salt water shriveled and became smaller. Why is that? Well, the answer is we do this a lot with foods. Foods are made from or come from living things. So whether we're talking about the fruits that we eat, whether we're talking about the vegetables, whether we're talking about the meats, when we talk about seasoning things, why do we add salt to our foods? The answer is that salt actually extracts water that would be found in the tissue from that living thing. Pepper adds a flavor where salt actually extracts water, leaving more flavor in whatever it is that we're eating. And so one of the things that cells need to navigate is the amount of salt that's in their environment. But this whole idea also emphasizes homeostasis, the ability for an organism to maintain a stable internal environment even when the outside environment is changing. And we see this specifically with like a lot of aquatic creatures. So examples of what organisms need to maintain homeostasis against would be salt, water, pH, temperature, just to name a few. And so the cell membrane is vital in making sure that the inside of the environment stays steady. Same thing with humans. Our skin helps protect us from the external environment, things like viruses, bacteria, and then also keeping our body temperature under control. This happens on a smaller scale with the cell skin, also known as the cell membrane. So within the cell membrane, there's 2 different types of transport, passive and active. And what we mean by these is passive goes from high concentration to low concentration, and that does not require energy. That is something that happens naturally. And so if I take a cup of water and I add a drop of food coloring, without shaking it or stirring it, if I was to wait, let's say, an hour or two, that food coloring would spread out. Things by nature want to be balanced. They want to be spread out. So that's why passive transport is a natural thing and also why it doesn't require energy. Contrary to that, active transport goes from low to high. So it makes it even more imbalanced. And because that is unnatural, it requires energy. Usually with cells, it's in the form of what we call ATP. The cell membrane is what we call permeable, which means that it allows some things to go through it for free, no problem. There's certain gaps in there where it's able to go through without any issue. However, other things it doesn't allow just to go through. It has to be monitored. So that's why our definition of cell membrane controls what comes into and out of the cell. So there are little gaps and there are little junctions in the cell membrane that will allow some things to go through no problem, where other things not so much. And that word is called permeability. So if something is permeable, it means that it can go through. So things like water, oxygen, carbon dioxide in some situations are allowed to go through without any consequence. Impermeable means that it cannot go through. So things that are able to go through, oxygen, carbon dioxide, sometimes water. Other things that don't go through would be like the big stuff. So big glucose molecules, larger food particles, and then also ions, which actually can cause not only molecules, but can also cause cells to become unstable. We actually use this to our advantage when we talk about nerve cells, which we'll talk about momentarily. So a concentration gradient means that you have a lot of something on one side and then not much on the other. And so what sometimes we see is we see an H with brackets that would be on this left side, and then an L with brackets over here. So things naturally will want to go down the concentration gradient and balance out. So if I had, let's say, 10 dots here and I had 0 dots here, we would want there to be about 5 dots and 5 dots on each side if this was a split type of scenario. So that's called a concentration gradient. So diffusion is anything that goes from a high concentration to a low concentration. This is the food coloring example, where I can put a drop of food coloring in a cup of water and it will naturally spread out without having to be shaken or stirred at all. Osmosis is more of a specific definition. Osmosis, is the movement of water only through a membrane from high to low. So if you're looking at the diagram, they're showing you here that there are these red dots, that's the sugar, the blue dots are the water, and so what will happen is because there's more water over here than over here, water will scoot over to this side and want to balance out. So you might have, for example, let's say 4 red dots for every blue dots. That's maybe the ratio of what might want to happen in this particular case. So osmosis is water specific and through a membrane. Diffusion is anything from high to low, but osmosis is more of a specific case that happens a lot within cells. And that's our example of the grape that we had started with. Other words that will come up in this chapter include what happens if we need to bring something into the cell that's fairly large. Maybe it's a hormone, maybe it's food, maybe it's a protein. We call that endocytosis. So endo, inside, cyto, cell, inside of the cell. And so what happens is that substance will push on our cell membrane, kind of create a little pocket or an envelope. The cell membrane will actually pinch onto itself and create a vesicle that then can be maybe taken to the rough ER, the smooth ER, the Golgi, and then be processed. So if endocytosis means to bring into the cell, exocytosis must mean to leave the cell. And we see that when we maybe excrete molecules into our bloodstream from a cell. Here's a micrograph of that actually taking place. So exocytosis means that you are getting rid of a substance or you're leaving the cell. And this happens with also same process with vesicles, except now the vesicle is actually going to join with the cell membrane and become part of it. Here is an illustration of that happening. So obviously pictures that look very, very similar. So this is showing you exocytosis. The vesicle will join with the cell membrane. It will actually stay there and now become part of the cell membrane itself. Where here, those molecules are pinching in, envelope is created, vesicle created, and then it might go to the Golgi body or some other part of the cell. All right, so what are some other types of diffusion or things that happen within the cell? So as we said, the cell membrane is permeable, which means that allows some things to go through, others not to go through. Well, how does that do that? Sometimes there are proteins that have a very specific tunnel that goes in and through them. So if the tunnel is this big, what it's saying is, okay, little circles, you can go through for free. Still high to low, we're still diffusion, high to low, still for free. It's just there's a protein there that's monitoring what can go through that channel or what can go through that particular gap. We can also see this with what we call tonics, where depending on if a cell is put into a salty condition, we know that the cell will shrink because water leaves the cell. If it's put into a isotonic solution, that just means that it's the same throughout. The water will stay the same size. If we put it into a pure water situation, also known as hypotonic, water will go into that cell because there's more water on the outside than the inside, and water again always wants to be equal on both sides. Facilitated diffusion, so still diffusion, still high to low. But what facilitate means, it means to help. So there's going to be a protein here that is just going to help things along. Again, the cell membrane is controlling what comes into or out of the cell. This time it's a type of what we call facilitated diffusion. The 2 slides ago, it was just letting it go through as long as it was the same size or a particular size. The facilitated diffusion is that it's going to recognize a certain shape. It's still going from high to low. It's just going to help it along. So it's recognizing, okay, it's okay if you come in. So the cell membrane, we sometimes call it the bouncer of the cell. Then we have those that are going to go opposite, or what we call active transports. And one of the most popular types of these that we have in the human body is called the sodium-potassium pump. So if you've ever wondered why we are able to react super fast, or why we are able to have reflexes, the answer is the sodium-potassium pump. So what happens here is that sodium and potassium are both pumped against their gradient. And so because that's unnatural, cell energy in the form of ATP is used. And this is a very, very big deal because, again, this is what allows us to react really, really fast. And this happens specifically on our nerve cells, where we are keeping not only the sodium wrong, but also the potassium wrong. And what I mean by wrong is I mean imbalance. So instead of them balancing out at, let's say, 3 on this side and 3 on that side, it's putting sodium at 6 on this side, 0 on this side, and it's putting potassium 6 on this side, 0 on the other. So they're both imbalanced. And what that does is it allows us to react really fast. So if you've heard of the knee-jerk reaction, or if you've heard of if you've ever gotten your pupils dilated, our pupils naturally get bigger or smaller depending on if we're in a super sunny situation or a dark situation. Our body is able to do that naturally because of that, what we call active transport, pumping those ions against their gradient when they're not supposed to. All right? And so in closing, vesicles, they help move things and they help make sure that things are balanced within those. We have receptor proteins that are going to be able to receive messages from some cells, and they will allow us to respond to our environment. That concludes our discussion on cell transport.